Operation Icebridge is a 6-year NASA mission that monitors the Earth's polar ice in the Arctic and Antarctic. It's the largest airborne survey of Earth's polar ice ever flown.
Beneath a kilometer of Antarctic ice, Mercer Subglacial Lake waits in total darkness, a world untouched by sunlight or fresh air.
It's possible that underneath the ice will be keys for us to actually save ourselves and reclaim our ancient past.
Scientists believed nothing could endure in such crushing isolation. Then, in one of the most daring missions ever attempted, a team drilled through the ice to find out.
The team is using a 3-kilometer-long hose to blast superheated sterile water through the ice.
If there are things buried under the miles of ice at Antarctica.
The lake was not sterile. It was alive, and the life they found should not exist by any standard we thought we understood. Antarctica's hidden mysteries. Antarctica has always been a land of extreme cold, blinding white ice, and almost total secrecy. Its sheer size and harsh conditions have prevented people from exploring much of it, and that lack of knowledge has fed the imagination for decades. One of the most enduring ideas has been that ancient civilizations might have thrived on Antarctica before the ice covered it. Writers and conspiracy theorists suggested that pyramids or other monumental structures were buried beneath the frozen surface, waiting to be revealed. Others went even further, arguing that alien bases could be hidden under the ice.
That Antarctica is some kind of alien base.
Rumors of extraterrestrial activity in Antarctica.
These claims often drew on blurry satellite images or unusual natural formations that were presented as proof. During the Cold War, Antarctica became the focus of even more speculation. Reports circulated that Nazi Germany had carried out secret missions there in the 1930s and 1940s. According to the rumors, the Nazis built hidden bases under the ice and developed advanced technologies that the world never knew about. While historians and scientists have shown that these claims were exaggerated or false, the stories stuck in the public imagination. They added to the view of Antarctica as a place of secrecy, where things too strange or dangerous for the outside world might be locked away. Another disturbing theory that circulated in the 20th century suggested that Antarctica was not only home to hidden bases or lost civilizations, but also a location for secret experiments involving biological weapons.
I 100% believe this is a spiritual war and that these people are working with dark entities, and that's what this is all about.
Some conspiracy theorists claimed that governments considered the frozen continent a safe place to hide dangerous research programs because any outbreak would be trapped under the ice. These ideas were never proven and were often dismissed as paranoia, but they added to Antarctica's reputation as a forbidden and unsettling frontier. Scientists, however, were not interested in alien bases, Nazi strongholds, or hidden laboratories. They knew that beneath the ice, there were real mysteries worth exploring. In the 1970s, British scientist Robin Bell and her colleagues began using airborne radar systems to map what lay below the ice sheet. Aircraft flying over Antarctica sent radar waves through the ice, and the echoes that came back revealed the shape of the land beneath. To the surprise of researchers, the signals also showed flat surfaces that looked like water, not rock. These were the first signs that entire lakes existed below the frozen sheet. Later, satellite missions such as NASA's ICES in the early 2000s confirmed that the ice above certain areas was shifting in ways that suggested water was filling and draining below. Together, radar surveys and satellites created the first real inventory of subglacial lakes. These lakes that had been hidden for thousands, and in some cases millions, of years. They were untouched by sunlight, shielded from the surface, and cut off from the rest of Earth's ecosystems. On the surface, it seemed impossible that anything could live in such a place. No photosynthesis was possible. Nutrients would be scarce. Oxygen would be trapped in only small amounts. Yet, these lakes existed, and their conditions raised an important question. Could life survive there? In time, that question would lead directly to a bold mission to drill into Mercer Subglacial Lake and find out the truth. The bold mission beneath a kilometer of ice. The project to reach Mercer Subglacial Lake was called SALSA, which stands for Subglacial Antarctic Lake's Scientific Access. This was not a single country's effort, but an international collaboration that involved the U.S. National Science Foundation, the Korea Polar Research Institute, and several universities and research centers. The name SALSA might sound lighthearted, but the mission was one of the most difficult scientific undertakings ever attempted in Antarctica. One of the leading figures was John Priscu, a polar microbiologist from Montana State University. Priscu had spent decades studying microbial life in extreme environments, including lakes in the Antarctic dry valleys. He became known as one of the world's top experts on how life can adapt to icy and nutrient-poor ecosystems. Alongside him were scientists from the University of Florida, the Desert Research Institute in Nevada, and research centers in South Korea. Each institution contributed different areas of expertise. Some focused on microbiology and genetics. Others specialized in ice sheet dynamics, glacial geology, or the engineering required to drill through ice without introducing contamination. The lake is not small. At the site where the drilling would take place, it stretched across an area of 143 square kilometers and is about 15 meters deep. For comparison, that is roughly the size of a large city. The preparation for this mission took years. It was not as simple as sending a drill through the ice. The team had to design a system that would keep the lake uncontaminated. If even a single surface microbe were introduced into the water, the results would be meaningless, because it would be impossible to know whether the organisms found were truly native to the lake or just surface contaminants. To solve this problem, they developed a special hot water drilling system. This system used water that was carefully filtered, sterilized with ultraviolet light,
The UV radiation will not have a residual impact on the water,
and treated so that it would not carry modern microbes into the lake. Beyond drilling, the team also had to plan transport and logistics. Antarctica is one of the harshest environments in the world. Everything needed for the project, from the drill to the fuel to the labs, had to be carried over the ice by tractors, snow vehicles, and cargo aircraft. The researchers set up a full camp on the surface above the lake, complete with sterile labs where samples could be analyzed immediately without being exposed to contamination. The challenge was not only to reach the lake, but to ensure that once the borehole was open, it stayed open long enough to lower their instruments and collect water and sediment. The borehole would naturally begin to refreeze, so timing was critical. Every step had to be coordinated and carried out with precision. This was more than just a technical exercise. The researchers were aware that they were carrying the responsibility of answering one of the biggest questions in Antarctic science. If life could be found in Mercer Subglacial Lake, it would reshape what scientists believed about the limits of survival on Earth. It might even provide clues about life on icy planets and moons elsewhere in the solar system. But as the team prepared, many questioned whether this ambitious and dangerous project could even succeed, especially since past attempts to drill into Antarctic lakes had often ended in failure. A history of failure, why many believed this mission would collapse. Whenever scientists set out to drill through Antarctica's ice in search of hidden lakes, the effort has been marked by frustration and disappointment. The most famous case was Lake Vostok. Lake Vostok is the largest known subglacial lake in Antarctica, buried under nearly 4 kilometers of ice. In the 1990s, Soviet scientists began drilling toward it, later joined by Russian researchers after the collapse of the Soviet Union. The operation was one of the most ambitious polar projects in history. The drilling used specialized ice core drills that relied on fluids such as kerosene and freon to keep the borehole open and prevent it from freezing shut. These fluids were effective in maintaining the borehole, but carried a major risk. If they leaked into the lake, they could contaminate its pristine waters. By 2012, the Russian team announced that they had finally reached Lake Vostok. The world watched with anticipation, but the excitement quickly turned to doubt. The first samples brought up showed signs of contamination from the drilling fluids. Many experts argued that the results could not be trusted. Instead of proving the existence of native life, the samples were labeled too dirty to prove anything. The controversy left Lake Vostok as an unresolved case, and critics warned that future projects risked repeating the same mistakes. In the early 2000s, another major attempt took place at Lake Ellsworth, a smaller subglacial lake in West Antarctica. A team of British scientists prepared for years, designing a hot water drilling system that would melt through 3 kilometers of ice. Unlike the Russian effort, their plan was built around preventing contamination by using clean water. When the operation finally began in 2012, hopes were high. However, within days, the project collapsed. The borehole began to close as the drill lost pressure, and the team was forced to abandon the mission without ever reaching the water. The failure cost millions of dollars and became a cautionary tale. If larger, well-funded projects like Vostok and Ellsworth could not deliver uncontaminated results, how could a smaller project like Mercer succeed? Scientists and policymakers were hesitant to believe that the SALSA team would achieve what others could not. The doubts went beyond logistics and technology. Some researchers questioned whether the effort was worth it at all. Without sunlight, without a steady source of nutrients, and with extreme cold, many believed that the lakes would be empty. To them, the search for life beneath the ice was chasing a fantasy. If their mission failed, it would not just be a setback. It would become another shameful entry in the long list of failed Antarctic drilling attempts. Despite this, the Mercer team pressed on, drilling into the unknown. The SALSA project began its fieldwork in late December 2018. Once the team created the borehole that pierced through more than a kilometer of ice, the next stage of the operation began, which involved retrieving samples that had been sealed away for thousands of years. This stage required a set of highly specialized tools to collect samples from Mercer Subglacial Lake without compromising their purity. Every instrument had to be carefully designed for both function and cleanliness, because the scientists knew that a single mistake could ruin years of preparation. One of the most important devices was called the Large Volume Water Transfer System. This machine was lowered into the borehole to collect and filter massive amounts of water directly from the lake. The system was able to concentrate microbial cells onto fine filters while still preserving their structure for later genetic analysis. The design of this system allowed the team to work with very low levels of biomass, which was expected in such an isolated and nutrient-poor environment. Another critical instrument was the sediment corer. Sediment corers are long cylindrical tools that can be lowered into the lake bed to capture layers of mud that have built up over centuries. These sediments are valuable because they contain a record of the lake's history. Each layer can reveal information about past ice movements, chemical changes, and possible microbial activity. The team used a device known as a multi-corer, which was capable of retrieving several cores at once. This increased the chance of success in case one sample was damaged during retrieval. To support these main tools, the scientists also used a sediment catcher attached to the bottom of the water transfer frame. This acted like a scoop, collecting loose particles as the frame was lowered and raised. It provided an additional source of material that could be tested for microbial life and chemical traces. Alongside these, bottles for water collection were lowered into the lake at different depths, allowing the researchers to compare conditions near the surface and closer to the lake bed. The work itself was exhausting. Antarctica's brutal weather repeatedly interfered with the mission. Freezing winds and snow slowed progress and threatened to block access to equipment. The borehole itself presented a constant danger because it began to refreeze as soon as the drill stopped circulating warm water. The team had to work quickly and in shifts, sometimes around the clock, to prevent losing access to the lake. They kept heated water constantly circulating near the surface opening, used insulated coverings to slow the refreezing process, and lowered instruments immediately after the borehole was ready in order to maximize the short window of access. Every piece of equipment had to be lowered and retrieved before the ice could close in. After about a week of non-stop drilling and round-the-clock work, the team finally succeeded. They collected uncontaminated water samples and retrieved cores of pristine sediment from the bottom of Mercer Subglacial Lake. Now came the real test. The samples had to be studied, and the results would determine whether the skeptics had been right or whether life had truly endured in the darkness beneath Antarctica's ice. Strange cells in the dark water. When the first samples from Mercer Subglacial Lake were brought to the surface and examined, the researchers immediately saw signs that the lake was not empty. Under the microscope, they detected microbial cells moving in the water. The temperature of this water was measured at minus 0.74 degrees Celsius, which is just below the freezing point of pure water, but still liquid because of pressure from the overlying ice. The fact that living cells were visible in such conditions stunned the team. Still, the scientists had to be sure that these cells were not simply contaminants from the surface. That was the greatest fear throughout the project. To answer this, they examined the genetic material of the organisms. The DNA sequences showed that these microbes were not modern intruders. Instead, they were genetically distinct and formed populations that had been isolated from surface ecosystems for a very long time. This confirmed that the life in the lake was native and not the result of contamination from the drilling process. Over the course of the analysis, the team identified a total of 3,170 microbial cells. Many of them did not match known organisms, which immediately suggested that Mercer Subglacial Lake hosted unique forms of life. Most of the cells belonged to two broad groups of microorganisms, bacteria and archaea. Bacteria are single-celled organisms found everywhere on Earth, from soils to oceans to the human body. Archaea are another group of single-celled organisms that are often associated with extreme environments such as hot springs or salt lakes. The presence of both groups in Mercer showed that the lake supported a diverse ecosystem, even though it was cut off from sunlight and modern nutrient cycles. The team also found evidence of viruses in the samples. Viruses cannot live independently, but their presence showed that the microbes in the lake were active enough to support infection and viral replication. This meant that the lake was not just home to scattered survivors. It was a functioning ecosystem with interactions between different forms of life. One of the most surprising results was that the microbes in Mercer Subglacial Lake were not closely related to marine organisms. Many researchers thought they might find ocean-based microbes because parts of Antarctica were once connected to the sea, and ancient seawater had reached some subglacial basins. Instead, the genetic comparisons showed something very different. The closest relatives of the Mercer microbes were species usually found in freshwater environments, such as lakes and rivers, and in soils. This suggested that their ancestors most likely came from surface lakes, or soils that existed before the West Antarctic ice sheet expanded and covered the region. When the ice advanced, those organisms were buried along with water and sediments, trapping them under the ice. From that point onward, they were cut off from the surface world. Over thousands and possibly millions of years of isolation, these buried microbes slowly changed and developed new traits that allowed them to survive in the extreme cold, darkness, and limited nutrient supply of the subglacial lake. The conclusion from these first observations was unavoidable. Mercer Subglacial Lake was not dead. It was alive with hidden ecosystems that had survived in complete darkness and extreme isolation for vast stretches of time. But the real shock came with deeper genetic analysis, which revealed that these organisms had powers and strategies for survival that no one had expected. Genetic aliens, new species born in isolation. Once the first signs of life in Mercer Subglacial Lake were confirmed, the researchers moved to the next stage of analysis. The key question was not only whether microbes were alive in the lake, but also what kind of microbes they were. To answer this, the team conducted genetic sequencing on single cells taken directly from the water and sediments. This was a highly precise method that allowed scientists to study the DNA of individual organisms instead of analyzing all of them together in a mixed sample. By doing this, they were able to identify 1,374 separate genomes. The results were extraordinary. When these genomes were compared with the massive databases of known microbial species, the majority of them did not fit into existing categories. Their average genetic similarity to known organisms was only 77%. For context, scientists use a 95% similarity threshold to classify whether two organisms belong to the same species. Anything below that threshold indicates a different species. This meant that nearly all of the organisms found in Mercer were completely new to science. Even more surprising, some of these genomes were not just different species, but so unusual that they could belong to entirely new categories of life at higher levels of classification. Scientists organize all living things in a hierarchy of groups called taxonomy. At the bottom of the hierarchy are species, which are the most specific level. A group of closely related species is called a genus. Several related genera make up a family. Families that share deeper similarities are grouped into orders, and above that are classes, phyla, and finally domains, which are the broadest divisions of life. To make this clearer with an example, humans and chimpanzees are classified as different species, but both belong to the same genus, which is Homo for humans and Pan for chimpanzees. Together, they are part of the same family of primates, which also includes gorillas and orangutans. That means even though humans and chimpanzees are different species, they are still quite closely related in the overall tree of life. What the Mercer genomes showed was separation on a much larger scale. Many of these microbes were not just distinct species. They were so genetically distant from anything ever studied that they could belong to new genera, new families, or possibly even new phyla. A phylum represents one of the highest levels of classification in biology. For example, all animals on Earth, from insects to fish to humans, belong to a single phylum called Chordata, if they have a backbone, or to other phyla if they do not. To discover microbes that might represent new phyla means that they are not just cousins or distant relatives of known species. They could be the start of completely new branches on the tree of life that had never been recorded before. The way these organisms clustered together in the genetic trees also told an important story. Instead of being spread randomly across known microbial groups, they formed distinct clusters of their own. This showed that they had been evolving separately from surface and marine life for long stretches of time. The isolation beneath the ice had created what was essentially an evolutionary laboratory. Without regular contact with outside gene pools, these microbes developed along their own independent paths. Knowing that the microbes in Mercer were unique was one thing, but another question remained. How could these organisms possibly survive without sunlight or a constant supply of nutrients? The answer was even stranger than the discovery of their genetic isolation. Survival without sun. The microbes inside Mercer Subglacial Lake do not have access to sunlight. Sunlight is the usual energy source for life on the surface of Earth, since plants and algae use it in photosynthesis to produce food. In Mercer, this process is impossible. The lake has been buried under more than a kilometer of ice for thousands of years, and no light penetrates that depth. For these organisms to survive, they had to rely on completely different sources of energy. The genetic analysis of their cells revealed how they accomplish this. The microbes gain energy by carrying out chemical reactions with compounds available in the lake. This process is known as chemolithotrophy. The term means eating rocks, but in practice, it refers to organisms that obtain energy by oxidizing inorganic substances rather than using sunlight or organic food. In Mercer, the microbes oxidize sulfur, iron, and nitrogen compounds that are present in the sediments or dissolved in the water. These reactions release energy that the cells can capture and use for survival. One of the most impressive findings was that some microbes can shift their metabolism depending on how much oxygen is available. When oxygen is present, they use it to fuel aerobic respiration, which is the same process humans rely on to generate energy. When oxygen is scarce, they switch to anaerobic processes that use alternative molecules such as nitrate or sulfate instead. This flexibility allows them to continue living even as the chemistry of the lake changes over time. Certain microbial groups were able to oxidize methane, a gas that builds up under ice as organic material slowly breaks down. This means the lake contains organisms that act as natural methane consumers. Without them, methane could accumulate unchecked and change the chemistry of the ecosystem. Others showed the ability to use carbon monoxide and fatty acids as energy sources. Some could even use compounds released by the grinding of rocks at the base of the glacier. This ability to harvest energy from multiple chemical sources shows how resourceful these organisms are in a place where nutrients are scarce. Among the most startling discoveries were microbes from a group called Pathescobacteria. These microbes had extremely small genomes, which means they carried only the most basic set of genes needed for survival. They lacked the genetic instructions to make many essential molecules, such as certain amino acids or lipids. Because of this, they could not live independently. Instead, they survived by forming close relationships with other microbes in the lake. Some of these relationships were cooperative, with Pathescobacteria sharing resources with their hosts. Others appeared parasitic, with Pathescobacteria taking nutrients without giving anything in return. This showed that survival strategies in Mercer went beyond simple chemistry and included complex interactions between different organisms. Viruses added another layer to this hidden world. Genetic evidence revealed that viruses frequently infected the microbial populations of the lake. These infections may have helped shape the evolution of the microbes by transferring genes between them or by forcing them to adapt to constant attacks. In this way, viruses were not just destructive agents, but also important drivers of diversity and resilience within the ecosystem. Together, these findings proved that Mercer Subglacial Lake is thriving in ways that were completely unexpected. What do you think about this discovery? Comment down below.